Welcome to another lesson by MedicoMedics, Learning Made Easy. Cell Biology, Chapter 2, Cell Structure and Organization. In this lesson, we will discuss different types of cells. We will delve deeper into the nucleus, discuss the mitochondria, ribosome, the endoplasmic reticulum, present the Golgi apparatus, or Golgi apparatus, present lysosomes and compare them with peroxisomes, present the cytoskeleton, discuss the cell membrane, and end with a summary. Now, when we talk about types of cells, like we mentioned in the overview in Chapter 1, we talk about prokaryotic cells and eukaryotic cells. And if you remember, prokaryotic cells were more simple, smaller, and they lack a nucleus. An example of a prokaryotic cell is bacteria. Now, the key components of prokaryotic cells is they have cell membrane, cytoplasm, DNA, and ribosomes. While eukaryotic cells, compared to prokaryotic cells, are more complex. They are larger. They have a nucleus and membrane-bound organelles. These are found in humans, animals, and plants. Now let's begin with the nucleus. Now, what is the structure of the nucleus? Well, it's enclosed by the nuclear envelope with pores for material exchange. It contains, so the nucleus contains chromatin, which is DNA and proteins, and the nucleolus makes ribosomes. Now, what's the function of the nucleus? Well, it stores genetic material, right? It houses DNA. Furthermore, it regulates cell activities by controlling gene expression. So the nucleus is like a boss's office where all instructions are stored and decisions are being made. Now let's look at how energy is created in the cells. When we talk about energy and the production of ATP, we talk about the mitochondria, known as the powerhouse of the cell. The structure is a double membrane. There is an outer membrane, seen here, and a folded inner membrane, also known as crista. Now, the mitochondria contains its own DNA and ribosomes. Now, the function of the mitochondria is to produce ATP through cellular respiration. And it plays a key role in energy metabolism and in programmed cell death, apoptosis. Now, in later courses and lectures, especially in biochemistry, we will delve much more in detail into the production of energy in the mitochondria. But right now, all you need to remember is that the mitochondria are like power plants. These generate the energy that our body needs to function. Now let's move to the ribosomes, which are the protein-making factories of the cell. Now let's talk about ribosomes, or protein factories. Their structure is made of RNA and proteins. They are found free in the cytoplasm or attached to the rough endoplasmic reticulum, RER. The ribosome and their function is to synthesize proteins by translating mRNA. Proteins are essential for cell structure and function. So the ribosomes are like chefs in a kitchen. They are cooking up proteins based on recipes from DNA. And if you remember, the DNA is housed in the nucleus. Now let's talk about another very important structure, which is the endoplasmic reticulum. The endoplasmic reticulum is like a factory assembly line where raw proteins and lipids are processed and packaged. Now we divide the endoplasmic reticulum into the rough endoplasmic reticulum, or RER, and the smooth endoplasmic reticulum, SER. Now, the rough reticulum is covered with ribosomes. It synthesizes and processes proteins. And you can see the rough endoplasmic reticulum here, and the ribosomes covering it. The smooth endoplasmic reticulum doesn't have ribosomes covering it, and it synthesizes lipids. It also detoxifies drugs and stores calcium. Now, once things are processed here, where do these products then go? And to learn more about the next step, we will now visit the Golgi apparatus. So the Golgi apparatus, or the shipping center, which should make sense since the endoplasmic reticulum was described as the assembly line, right? We started with the nucleus containing DNA. We then had ribosomes reading the mRNA and translating proteins and producing proteins. These then continue into the endoplasmic reticulum, into the assembly line, quote unquote, and then arrive at the Golgi apparatus, the shipping center. So the Golgi apparatus can be thought of as the post office, where packaging and shipping of molecules are made and then decided on where to go. So the structure of the Golgi apparatus is that it contains stacks of membrane-bound sacs, or cisternae. Its function is to modify, sort, and package proteins and lipids for transport. It also produces lysosomes and secretory vesicles. And the Golgi apparatus is illustrated here. Let's now examine the cell's recycling and waste disposal system, its lysosomes. So we've reached the Golgi apparatus where packing and sorting and shipping has been done. Now we need to have a system in place where waste is broken down, right? So lysosomes contain enzymes that break down waste and cellular debris. It also plays a role in autophagy, meaning recycling damaged organelles. Compared to that, we have the peroxisomes that break down fatty acids and toxins. It contains enzymes for oxidative reactions. So lysosomes can be thought of as the cleanup crew, and peroxisomes are like detox centers. Now, how do all these organelles that we have talked about, the ribosomes, the endoplasmic reticulum, the Golgi apparatus, how do these organelles stay in place? To find out, we need to talk about the cytoskeleton. Now, the cytoskeleton is the cell's framework. Its structure is a network of protein filaments, so microfilaments, microtubules, and intermediate filaments. What is the function of the cytoskeleton? Is to maintain the cell shape. It's to facilitate intracellular transport and cell division. Furthermore, another function is that it enables cell movement. So what the cytoskeleton does is that it provides the cell structure and furthermore provides transport routes. Now let's finish by understanding how cells interact with their environment through the cell membrane. We have now arrived at the cell membrane, which is made of a phospholipid bilayer with embedded proteins. It is dynamic and semi-permeable. What is the function of the cell membrane? Well, mainly to control what enters and what exits the cell. Furthermore, it facilitates communication through receptor proteins. Now, as we move on into further lectures, we will delve into details of how this cell membrane is constructed, how things from the outside actually enter into the cell, why things from the outside leave the cell, these different channels whereby things are entering, and also the benefits of having a phospholipid bilayer. Because what you will learn is that the outer surface of the cell is hydrophilic, while the inner part of the membrane is hydrophobic. And this leads to different considerations to keep in mind. But more on that as we move on into further lectures. And now, in summary then, the cell is a complex structure with specialized organelles. Key organelles to remember from this overview lesson include the nucleus, ribosomes, the mitochondria, the smooth endoplasmic reticulum and the rough endoplasmic reticulum, Golgi apparatus, lysosomes and peroxisomes, cytoskeleton, and cell membrane. And if you remember what sets them apart, in the nucleus is where we house the DNA. The ribosome is our protein factory. We have the mitochondria, the powerhouse where energy is produced. We have the smooth and the rough endoplasmic reticulum, where we have the synthesis of lipids and proteins. Furthermore, moving along into the Golgi apparatus, where we have the packaging, sorting, and shipping. And then we have the recycling part of the cell, the lysosomes and the detoxification centers in our peroxisomes. What gave the cell its structure and what helped facilitate transport routes? Well, we talked about the cytoskeleton. And then, at the end, what is it that actually allows for things to enter into the cell or leave the cell? These are determined by our cell membrane, which was made by a phospholipid bilayer. Now, each organelle plays a distinct and vital role in maintaining life. Understanding cell structure is fundamental to diagnosing and treating diseases. Now, let's move forward to the next chapter where we will explore how substances move in and out of cells, meaning we will go deeper into the cell membrane. And that's the end of this lecture. Continue now to chapter 3.